Welcome to Cell Biology. Today, we are going to explore one of the most fascinating structures in all of biology, the plasma membrane. Imagine a medieval castle protecting a bustling city. The castle walls determine who gets in, who gets out, and how the city communicates with the outside world. In biology, every single cell has its own protective wall, called the plasma membrane. Because it is incredibly thin, scientists had to play a historical detective game to figure out its structure. Let us dive in and uncover how we built our understanding of the cell's outer boundary. Now that we know what the plasma membrane does, let us look at how we study it. A plasma membrane model is a theoretical representation that describes the structural arrangement of lipids, proteins, and carbohydrates forming the cell boundary. Historically, several models were proposed as microscopy and biochemical techniques advanced. We started with the primitive lipid bilayer model by Gorder and Grendel, moved to the flawed Davson-Danieli sandwich model, and finally arrived at the currently accepted fluid mosaic model. Why do we care? Because the plasma membrane is the absolute gatekeeper of life. If we do not understand how it is built, we cannot understand how medicines enter cells, how viruses infect us, or how our muscles contract. Building on that historical context, let us examine the very first major breakthrough. Even though biology seems like it is all about memorizing parts, early membrane models were actually discovered using simple geometry and math. In 1925, scientists Gorder and Grendel performed a brilliant experiment. They extracted the lipids, or fats, from red blood cells and spread them out on a water surface. Their goal was simple but profound. They wanted to compare the surface area of the extracted lipids to the surface area of the original intact cells. By doing this comparison, they hoped to figure out exactly how many layers of fat surrounded a single cell. To make sense of their experiment, Gorder and Grendel used a straightforward mathematical relationship. Conceptually, this formula compares the total footprint of the fat to the total footprint of the cells it came from. Let us break down the symbols. R represents the ratio of the lipid surface area to the cell surface area. A sub L stands for the total surface area that the extracted lipids cover when spread flat on water, measured in square micrometers. A sub C represents the total calculated surface area of the original red blood cells, also measured in square micrometers. If the amount of lipids increases while the cell area stays the same, the ratio R would increase, indicating a thicker or multi-layered membrane. Before we move on, let us see this with a concrete example. A scientist extracts all the lipid molecules from 1,000 red blood cells. We are given that a single cell has a surface area of 100 square micrometers, and the extracted lipids cover 200,000 square micrometers. We need to calculate the ratio. First, we find the total surface area of all original cells by multiplying 1,000 cells by 100 square micrometers, giving 100,000 square micrometers. Next, we divide the total lipid area, 200,000, by the total cell area, 100,000. The math simplifies perfectly to a final answer of 2. This physical result means there is exactly enough fat to wrap around the cell twice. So we have proven that the ratio equals 2. This brings us to the mathematical proof of the membrane structure. What this chunk of proof is establishing is the physical interpretation of our mathematical result. When the ratio is exactly 2, it means the surface area of the lipids is double the surface area of the cells they came from. The mathematical reasoning here is simple geometry. If you have enough material to cover a sphere twice, that material must be arranged in two distinct layers. This intuition is exactly how humanity discovered that the cell membrane must be a lipid bilayer, meaning it is two layers thick. This foundational proof connects directly to the more complex models that followed. Now that we understand the basic bilayer, let us move to the modern understanding of the membrane. Imagine a crowded swimming pool filled with thousands of floating ping pong balls. If you drop a few large beach balls into this pool, they will float among the ping pong balls, bumping around and changing positions. The plasma membrane works exactly like this. Proposed by Singer and Nicholson in 1972, the fluid mosaic model states that the plasma membrane is a fluid combination of phospholipids, cholesterol, and proteins. The ping pong balls are the lipid molecules, forming a liquid-like sea, and the beach balls are the proteins floating within it. It is not a solid, rigid wall. It is a constantly moving, flowing liquid boundary. This dynamic structure we just visualized leads us to an important question. Why do we use the words fluid and mosaic? The word fluid refers to the fact that the lipid molecules can move laterally, or side to side, within their own layer. This means the membrane is flexible. The word mosaic refers to the patchwork arrangement of different proteins embedded within the lipid bilayer, much like tiles in a mosaic artwork. These proteins perform the actual work, like transporting molecules. This model completely revolutionized biology. If the membrane were a solid crust, a cell could never divide into two during mitosis without shattering. Furthermore, the fluidity allows membrane proteins to drift and interact, which is absolutely essential for cell signaling and hormone reception. To truly grasp this concept, let us look at a detailed visual representation. Here, you are seeing a cross-section of the fluid mosaic membrane model. Notice the main structure, a double layer of lipids with their round hydrophilic heads facing outward and their stringy hydrophobic tails pointing inward. This is the bilayer Gorder and Grendel proved. Embedded within this lipid sea, you can see large integral proteins that span the entire width of the membrane, acting as channels. On the surfaces, there are peripheral proteins attached. You can also spot cholesterol molecules tucked between the lipid tails, and carbohydrate chains like glycolipids and glycoproteins extending from the outer surface. This diagram beautifully captures the dynamic, two-dimensional liquid nature of the cell boundary. Having seen the fluid nature of the membrane, we must ask, how do we actually measure how fluid or liquid-like it is? Scientists use a brilliant technique called fluorescence recovery after photobleaching, often abbreviated as FRAP. In this method, researchers attach glowing fluorescent tags to membrane proteins. Then, they blast a small, specific area with a laser to destroy the glow, creating a dark, bleached spot. Finally, they time how long it takes for new, unbleached glowing proteins to float into the dark spot from the surrounding area. By measuring this recovery time, we can calculate exactly how fast the molecules are swimming through the lipid sea. The FRAP experiment we just discussed gives us the data needed for this mathematical formula. Conceptually, this equation translates the time it takes for a spot to recover into a standard speed of movement. Let us define the symbols. D represents the diffusion coefficient, which measures the speed of the lateral movement of molecules in square micrometers per second. The variable W represents the radius of the laser beam used to bleach the spot, measured in micrometers. The variable T sub 1/2 represents the half time of recovery, which is the time in seconds it takes for the bleached spot to recover half of its original brightness. If the membrane becomes more fluid, perhaps due to a higher temperature, the proteins move faster. This causes the recovery time to decrease, which mathematically causes the diffusion coefficient D to increase. Now that we have our formula, let us apply it to a real laboratory scenario. A researcher is studying a new integral membrane protein. We are given that the laser beam has a radius of 2 micrometers, and it takes 10 seconds for the spot to recover half of its original fluorescence. We need to calculate the diffusion coefficient. First, we square the radius of 2 micrometers, which gives us 4 square micrometers. Next, we multiply the half-time of 10 seconds by the constant 4, giving a denominator of 40 seconds. Finally, we divide 4 by 40. This gives us a final answer of 0.1 square micrometers per second. This physically suggests the protein is moving relatively slowly, meaning it might be very large or anchored to the cytoskeleton inside the cell. Now that we have used this formula, let us understand where its specific components come from. What this chunk of proof is establishing is the physical geometry behind the math. In our first step, squaring the radius, W, relates directly to the total area of the dark spot. It represents the spatial distance the molecules must cover to restore the glowing color. In the denominator, multiplying the half-time by the constant 4 is not arbitrary. The mathematical reasoning here is that this constant accounts for the two-dimensional random walk nature of the proteins diffusing through the flat membrane. It scales the time to match the geometry of the diffusion process. This intuition helps us see that the formula is perfectly tuned to the two-dimensional liquid nature of the fluid mosaic model. We have covered a lot of ground today, so let us review the most important ideas. First, the plasma membrane is not a solid wall, but a highly dynamic two-dimensional liquid where lipids provide structure and proteins perform vital functions. Second, we saw how Gorder and Grendel used simple math to prove the existence of the lipid bilayer. Third, we explored the Singer-Nicholson fluid mosaic model, which explains how cells can change shape, divide, and communicate. Finally, we learned how the FRAP technique mathematically measures membrane fluidity. Understanding this fluid, mosaic nature is the absolute foundation for studying how cells survive in a changing environment. You have now mastered the core models of the plasma membrane: